Thank you, Jay Varma from Africa CDC. It was a great talk, and I'm a little bit terrified because I sort of enjoy hate reading things on the internet about superfoods and how your diet will boost your immunity, and now I may be wrong.
That'd be terrible.
So should we all follow Gwyneth Paltrow and et cetera instead?
Yeah, it'd be terrible if press accounts of superfoods were wrong. I mean, that'll be a big surprise to everyone. What I didn't talk about here is there's a lot of emerging research that, so microbiome research and its intersection with public health advice about diet is twofold. So the first piece is that we're getting mechanistic understanding of a lot of public health recommendations that are very solid and evidence-based. Like the reason you should eat more fiber is primarily for butyrate production in the gut from the microbes that ferment fiber. We know which ones they are and how they work. And we can also start to talk about counter exceptions like people with IBD who should be staying away from the fiber because for them it's harmful. The second thing that's much more exciting and that's coming is personalized recommendations that have very large effects. So part of the reason why the supermarket tabloid diets don't work is they found one person who loses, say, 20 kilos on that diet. But if you try it on 1,000 people, some of them are going to lose 20 kilos, some of them are going to gain 21 kilos, and overall it's a wash. So if you think of it as being kind of like, say, blood transfusions, in the early days of blood transfusions, it could save your life, but it had a good chance of killing you. But then once blood types were discovered, blood transfusions became a miracle. And so the question is, can we discover a relatively small number of types of microbes in the gut so that the public health recommendation can be, what you should do is you should get a test and then you should take a discrete evidence-based step based on that test. And that, I think, is where it's going, although the data isn't in yet to change any recommendations right now. But certainly the data, not just on things like fiber, but on things like artificial sweeteners and emulsifiers, both of which there's mechanistic stories about how they're bad for your gut microbiome, is looking both very interesting and consistent with policy recommendation, and also the individual effect sizes are large. So maybe you would do just fine on artificial sweeteners, but the person sitting next to you wouldn't. And then the same sort of thing's true for red meat, where whether the carnitine and choline in it get converted to trimethylamine and then onto TMAO and cause atherosclerosis depends a lot on the details of what bacteria you have in the gut. Hi.
When people talk of epigenetics mostly, they talk about epigenetic changes in the gene expression of human genes. Now, what is the nature of epigenetic changes in the microbiome genes? And does it really have a fairly long-term effect? Apart from changing the colony character, are the environmental exposures more likely to also lead to epigenetic changes in the huge genetic pool that the microbiome represents?
Yeah, great question. Very little is known about the epigenetics of the bacteria themselves. They don't have chromatin, obviously. But they do have methylation, and there's some intriguing evidence of this epigenetic state in the bacterial genomes themselves. Another interesting thing is a lot of bacterially produced metabolites, notably butyrate, which is a histone deacetylase inhibitor, have effects on the host epigenome. And just in the last year, there have been about half a dozen papers showing very strong links between what microbes you have and host methylation and histone modification markers. So it's very much an emerging field, but it's a very exciting one. What is slowing it down is, in general, the labs that study the microbiome aren't the same as the ones that study epigenetics, and both of them require a complex technology stack that's both difficult and expensive. So funding projects at the level where both of those techniques can be combined in the same project has been relatively challenging, but it's really important. And then adding on things like metabolite profiling as well, so you can understand what small molecules are leading to those epigenetic changes, and then profiling different tissues around the body. So when we change the microbiomes of mice, we see gene expression changes in the brain, for example. All of this stuff is going to be really important to combine to get a systems level understanding.
Yeah, that was a really fascinating talk.
Thanks.
Now, a lot of the interest and medical interest in this area has to do with the interplay between the gut microbiota, the gut epithelium, and the immune system.
Mm-hmm.
Yet most of the measurements and comparisons use stool samples. I was wondering if there's data showing that that's backed up by biopsy samples or sampling that happens closer to the epithelium than sort of the end product.
Yeah, so great question. We've done that on a number of projects, including the Givas et al. Cell-hosted microbe paper on the risk cohort. So in general, for biomarkers, you can get good biomarkers out of the stool in most cases, although sometimes they're not as good as having a biopsy specimen. If you want to look at mechanism, there are taxa that are important at the mucosal layer that will not come out in the stool. So stool is much more useful for biomarker studies than for mechanistic studies. And when you go to get to mechanism, you want to use sacrifice mice where you have as much tissue as you need, and you want to do biopsies in humans. One important consideration is longitudinal studies. So we're seeing that variation in the microbiome itself, even on a daily timescale, is very important for predicting future state of immunologically linked phenotypes, like IBD flare, for example. And so you're definitely not going to scope your patient every day. So figuring out what we can get from stool while recognizing its limitations is really important. But while it's limited, it's certainly not completely useless.
Thank you, terrific. Many non-antibiotic pharmaceuticals also have antibacterial activity, and some of these are taken for chronic conditions. How comprehensively has that been looked at?
Yeah, thank you for asking me that. That's one of the things I dropped to fit this talk in 30 minutes, so I appreciate it. Just in the last 2 years, Pierre Bock's lab and Pete Turnbell's lab at UCSF, which you're probably thinking of, and 3 or 4 others have discovered that a huge number of pharmaceuticals that are not traditionally considered antibiotics, everything from metformin to antipsychotic drugs to SSRIs, have large impacts on individual gut microbes and also on the consortium if you take a fecal slurry and you incubate it. We know almost nothing at this point because only a few dozen compounds have been tested. They've only been tested against a limited range of microbiomes, so they haven't been tested in non-Westerners. They have not been tested in children. And even more shockingly, we have no idea how those pharmaceuticals interact with the environment, whether you're talking about the microbiomes of fish or the microbiomes of seawater or the microbiomes of the soil where you're using sewage as an input to agricultural crops and what those downstream effects are. So research in this area is very urgently needed. Another related issue is microplastics, where there are just 1 or 2 studies looking at the impact of microplastics on the microbiome, but by just providing a different kind of surface that microbes can attach to, the strong evidence from Justin Sonnenberg's lab, for example, that microbes love to attach to fibers, and that spatial structuring modifies their activity in the gut a lot, and it's very likely that the same is true for microplastics and other non-nutritive things that we're ingesting.
Kevin Oliver from EcoHealth Alliance. Very interesting talk, Rob. Thank you. I have a question about the role of viruses, particularly bacteriophages, in shaping some of the human microbial communities. Thinking of some of the recent work that's been done looking at carbon cycling in the ocean, how it's had such a dramatic influence globally on that. So do you see evidence of like epidemics throughout the microbiome happening with, or is it so diverse that it's resilient to those?
Yeah, great question. So again, I dropped that in the interest of time. You cannot see phages with 16S ribosomal RNA profiling, but shotgun metagenomics is becoming increasingly prevalent, and so you're seeing more and more phage studies coming out now from Forrest Rower's lab, from Jeff Gordon's lab, from Rick Bushman's lab, and from a number of others. The prospect of understanding phage bacterial dynamics is very exciting, but you need large sample sizes and you need very dense time series, and that in general has not been done yet just because of the cost of shotgun metagenomics. But with the NovaSeq S4 flow cells, that drops the cost dramatically even from what it was last year. So I think you will see a lot more of those kinds of studies in the near future. The other thing that's really exciting is the potential for phage therapy. I mentioned very briefly IPATH, the new phage therapy center we have at UC San Diego, and so that has been remarkably clinically effective for patients who have been written off completely by infectious disease specialists as a last-ditch measure under IND for compassionate use. So I think understanding the phage, learning how to use them to modify our microbiomes intentionally is very exciting. And given that you can do a total synthesis of a phage, so you can just type the sequence into your computer and order a phage with exactly that sequence, the potential to do it in a designed manner, I think, is very high. Although combining the phage engineering side with the microbiome side, again, it's different communities that need to be brought together to make progress.
Great, thank you.
Sort of a related question, which is looking beyond bacteria as part of the microbiome. So viruses, fungi, is there a better understanding about what that total complex might look like and what the implications might be?
Yeah, absolutely. So, so in Westerners, you'll pick up a few species of fungi as well, mostly Candida and Saccharomyces and a few others. In non-Western populations like the Hadza, the Yanomami, and so on, we see a much more diverse repertoire of fungi, and also we see a lot more microbial eukaryotes of other kinds, like protists and so forth. In general, the move of the whole field towards shotgun metagenomics is going to completely revolutionize what we know about everything except the bacteria and the archaea, which are the only things picked up efficiently and with decent taxonomic resolution by most amplicon studies. However, a key limitation is the reference databases. So in order to take shotgun metagenomics, what you're getting is a lot of short fragments of DNA, a couple of hundred bases long, and you need to match them up to a reference database to make sense of it. And we just don't have the reference databases that we need, especially the viruses and the fungi. But even the type strains, it would cost something like $4 million to sequence every type strain of bacteria, but no agency has picked it up yet. And it's the kind of thing that's outside the budget of any individual lab. But it's also, because it's interdisciplinary, it falls between the mandates of different agencies, and then getting, say, the USDA to pick up the soil ones, the NIH to pick up, I guess you'd have to get NIAID to pick up the specifically pathogenic ones. There's no particular mandate for the non-pathogenic human-associated microbes because NHGRI is not interested in that. You see it rapidly becomes a patchwork of frustration trying to get the agencies to work together. But just sequencing all the type strains and then sequencing as many fungi and protists as we can get our hands on from human and animal stool isolates to put them in context, that would be an amazingly valuable resource. It would greatly increase the efficiency of all metagenomic studies.
Yeah, I have a question. Rima Kabaz from CDC. Thank you. A great, very stimulating talk. I wanted to go back to the 1% human genes that we carry, and in terms of disease causation, I mean, you make a great case, and I'm a believer the role of microbiome. But so how do you factor that? And are there studies looking when you look at causation and look at, then moving to risk and personalized recommendation? Are studies incorporating host as well as microbiome?
Yeah, great question. There are some studies and an increasing number incorporating both host and microbial genomes. I showed you the one from Erin Segal's lab where they showed that the microbiome associations with phenotype were much greater than host. That has also been seen in other similar studies, like the one we did with Tim Spector and Ruth Lay in the Twins UK cohort, and in a number of others. In terms of personalized recommendations, we're not quite there yet. What we know is that individual differences are very large, but we don't have a test where we could take your sample and integrate it into that data frame. And that's something where the input of agencies like the CDC, the FDA, NIST, and so on, to develop standards where you could be confident that your individual sample could be integrated into those reference ranges and data frames, that would be extremely helpful, but that doesn't exist yet. And again, that's something that will need to be retrained in different cities within the US, in different parts of the world, because the reference ranges are going to be completely different. So, so when I was in Rome yesterday, one of the delegates at FAO took me aside after my talk and unfolded his microbiome profile, and that he'd got from a testing service there, and it said that he had very little Akkermansia because he had 1% Akkermansia and the reference range was 37% Akkermansia. And I have never seen an individual with 37% Akkermansia in looking at tens of thousands of individuals. So I have a lot of questions about what the test was and what the reference range was, right? But we really need to either standardize this stuff to make it useful at the individual level, or find a way for people to integrate themselves into a particular data universe that's useful, rather than just having these printouts that are completely uninterpretable, which is kind of the state of play at the moment.
Well, thanks.
Thanks again.
Heroic stuff. Running through the streets of New York is in itself. And I wonder what that's done to your microbiome. That was going to be my question. It may actually improve it that you went quickly through the streets of New York. Thank you for that. What is actually known as too much information. Okay, so it's my joy to sum up this afternoon briefly, and all I can say is, wow, it's a scientific and emotional roller coaster ride for me. And one of the great pleasures of the former microbial threats is that you learn a lot. You know, you're always being stretched, even if it's your field. This is not my field, and I knew I was going to learn a lot, but I'm just completely overwhelmed by what I've actually taken in. And I mean, it's gone from everything from the really shocking that the most disadvantaged people that we heard this morning have the highest rates of multi-morbidities, and that's highest in the young. So this really does get to equity. So that's the first emotional significance, that we really need to shift the health paradigm to understand the broader impact. And health doesn't just mean I'm sick and I need a drug. It means I need to eat the right food, I need to do the right exercise, I need to live in the right place. And this is an interesting holistic view. And we heard that from both talks this morning about getting into the syndemic side. And I really enjoyed the question from Marcus and then the answer about complexity. Clearly, you asked that question too soon, because look at the complexity now. And the answer's still going to be the same. We can't just simplify it. We're going to embrace it, understand it, absorb it, then hopefully get something out of it that we can actually use in a public health setting. But I think what's been interesting, from this afternoon, going through the case studies and finding out that, even more complexity if we look at the microbiome and food allergies, the Alzheimer's and gingivitis. What's interesting is the complexity increases, but now we start to see some actual treatments or actual therapies that do simplify things. And obviously, the mechanism is going to be ridiculously complex and poorly understood right now. So my take-home message, one of the things I learned, and I often, you shouldn't do this, but it's kind of human. You sit there and you listen to these talks and you think, well, what should I be doing tomorrow that will make my microbiome better or my risk of food allergies less? Should I be eating high fiber or should I be... One thing I've learned is, our little dog, I think it's okay that I let it lick my face, but what happens to the dog, the poor thing? That's one message for me for tomorrow. But I also was very interested from the food allergy point of view. The irony of all of this, because we're still falling into that other human trap, which is clearly one of the reasons why we've got food allergy issues in our modern society, is because we don't live in the traditional state which we've evolved into. The lifestyle change has driven that. The solution that's proposed is then to create a synthetic peptide which we'll market and take. And it's kind of interesting to me. I know that that we're too far along the path of urbanization and modernity to go back. But it does talk to this idea about superfoods and organic foods, where the mechanism's not known, but people are doing it, and you think, there must be something in it, and vegetarianism. These things where people are out there trying to grab to something healthy. And, there is a dark side to this as well, and I think for just about every talk today, there must be some sort of quack solution that I could purchase somewhere along the way. The solution would be, give me some of those bacteria, and how much is that going to cost? I need that peptide, or I need my profile. So there's a lot of room for abuse of that too. So there's a lot of complexity to deal with. But Rob, you really nailed it at the end by just whacking us with the most ridiculous dose of complexity we've had yet. It's nice to know that you don't understand some of the mechanisms as well, and that there's much more out there, much more to come. And I buy into the idea that we will look in a mirror and get this one day, and it's coming sooner than we think if you look at the accelerating pace of technology. But to me, the biggest thing that you said today, which wasn't that, which is a little bit cryptic. You even changed our understanding of Darwinian evolution to Lamarckian evolution. You showed that there's an inheritance of acquired characteristics in one of your slides, that your mother's microbiome, you pick that up. And if you shift that, you can pick that up too. And you can transpose that microbiome to another organism and it can acquire those characteristics. That's absolutely mind-blowing. Lamarck would be turning over in his grave and leaping out for joy right now. So that's the start of this 1.5 day workshop. That's half a day of it. So we've got some complexity. And by the way, Dennis, the Global Virome Project, forget about it. This is the real complexity here. That one's going to be easy and less expensive. So tomorrow we're going to try and understand this complexity. We're going to grasp blindly around this outrageously huge elephant and try and understand what's going on and trying to think through future trends and trajectories and solutions to where we can use this incredible wealth, this wave of information that's coming, and turn it into public health good. So I want to thank all the speakers again for an amazing first afternoon. Thank you. And I've just got a little bit of housekeeping. We have a reception outside. Then some of us have dinner. But before we go, can I ask you to leave your badges and cards on the tables? We'll be back in the same room. The workshop will start here at 8:30 tomorrow morning sharp. And please leave them there. And don't worry about your name badge. It's a good talking point when people don't know your name. It breaks the ice. So thank you very much. Let's go and have a glass of wine and try and understand this stuff.